Okay. Hello, everybody. Can I have your attention, please? So today we are starting arguably one of my favorite topics. I know I always say that, but this one is really one of my favorite topics. And we're gonna so for the next few lectures we're gonna talk about cytoskeleton, kind of quite a bit of nitty-gritty information, borderline biophysics, and then reaching out to actual cell biology, which is the reason why you're here. And then we're going to build on these cytoskeletal structures or lectures to talk about cell polarity and cell migration, which are more integrated processes. And because we missed one class, I mean, I guess you still have to figure out if we're going to squeeze in a cell adhesion lecture or when it's going to happen. Anyway, so jumping into this topic, not so fast. So I'm going to start easy with just a few illustrations of what the process is about. So we've been talking about cells, and with me, we've been talking about membrane-bound compartments, lipids, and so on. And it was clear throughout that there was some structure, some organizations, but lipids themselves are not the most mechanically reliable structures or elements in the cell. A lot of the cell structure depends on the cytoskeleton. So that's why the name itself implies that. That's the skeletal of the cell. And here you're just looking at two different examples of classic dichotomy of cellular organization and epithelial cells. So that's really closed-up view of microvilli in an enterocyte. So we're going to see those cells again and again. We've already seen that. When did we see that?
Polarization of the cell.
Polarization of the cell, and we talked about trafficking, right? How certain vesicles should be sent one way or another. But the trafficking itself, I already alluded to the idea that for vesicles to move, you need that's mechanical force, so you're going to need molecular motors. Some of them we're going to see today, myosins. And you're also going to need tracks for these molecular motors to move on. And some of those we're going to talk about today. It's actin. And microtubules we're going to talk after the spring break now. But on the other hand, so this snapshot makes it look like it's stable over time, so maybe not so much dynamics. The reality is that the cytoskeleton is always in kind of a steady state. Even when it looks stable over time, at the scale of the cell, you have constant replacement of elements. And when the cell is actually migrating, so we're going to see that. So this is here a snapshot of a cell. So the cell body is here with the nucleus, and you see these large protrusions. So there's a lamellipodium, there are filopodia, and so on. This is, of course, extremely dynamic, and I'm going to show you some movies in future lectures. So that dynamic is also supported by the cytoskeleton, and an essential aspect of the cytoskeleton, again, is this dynamic, the ability to make and break cytoskeletal elements in a spatially and temporally regulated manner. So we're going to spend quite a bit of time talking about it. So in cartoon forms, almost kind of repeating myself, what I just told you is depicted here. So you may have epithelial cells, right? So tightly adhering with each other. That's the key difference, right? Epithelial cells are engaged in cell-cell adhesion. There's a lot of coherence, mechanical coherence in the tissue. And in particular at this level, and for enterocytes, this area here, these junctions, these cell-cell junctions here, or sometimes called adherence junction, they are basically hermetic to, they don't let anything go in between cells, right? So that's part of what I was telling you. You have clearly separate apical domains and basolateral domains. So we talked about it about vesicles, but in terms of cytoskeleton, you also have key differences. Here in the microvilli, you have bundles of actin filaments, whereas on the other side, at the cortex, usually you have more of a meshwork of actin filaments. Okay? So today I'm going to tell you a great deal about how this is established. And in migratory cells, Instead, the organization of the actin cytoskeleton is different from the polarized epithelial cells. In the lamellipodium, you have a very dynamic meshwork, and we're going to see that over and over again. Elsewhere in the cell, you have more stable stress fibers. All of those are made of actin, right? So it's one of the questions we're going to have to answer, how using the same molecule, you're creating this diversity of cytoskeletal features. And those cartoons depict the other elements. So microfilaments are actin, the red ones. We're going to talk about microtubules. The organization is quite different, and I'm going to emphasize the differences throughout the next few lectures. And briefly, next time, we're also going to touch upon intermediate filaments, which are more stable, somewhat less dynamic structures. Now, taking a closer look at these actual components, again today, so little by little, we're going to go into closer, and we're going to take closer and closer look at these cytoskeletal elements. So microfilaments, exclusively made of actin. I'm going to tell you there are a couple of flavors of actin, but by and large, we're going to focus on beta-actin, kind of the main one. They are always thin and, of course, elongated. So they always have this sort of fibrous nature. But that's the microfilament. And as we'll see, it's a polymer. The protein itself is not a fibrous protein. Microtubules have the same kind of logic. They tend to make fibers, actually tubules, hence the name. The subunits are these alpha and beta tubulin, and they are slightly thicker and longer, as we'll see, and they really make a tube. So I'm not going to discuss them a lot today, because that we're going to emphasize those next time around. And finally, intermediate filaments, also fibrous. Now the big difference between actin and tubulin and these intermediate filaments, so those are many different kinds of proteins. The monomers are themselves fibrous. They just make higher order assemblies, and those are much less dynamic compared to what we're going to see today about actin, and next time about tubulin. Okay? So let's focus on actin microfilaments. Again, I'm still in my kind of introductory slide, so this sort of bird's eye views. So yet again, migratory cell, this time stained with a compound called phalloidin. It's classically used to visualize filamentous actin. It's a toxin from a fungus, and you can make it in vitro fluorescently labeled. So when you treat your cells, or when you stain fixed cells with phalloidin, you can see the actin filaments. Importantly, you see the filamentous form of actin, or F-actin, not the monomers, the globular form of actin, or G-actin. And I'm going to get back to that again and again. So what you see here is, and I already said that, some organization. So actin is everywhere. You agree with that? Yes. But it looks different in different places. At the front here, you have these short spikes. More towards the back, you have this much longer stress fibers. So these short spikes are filopodia. And here at the leading edge, you have this staining here, which is more diffuse, and that's the meshwork. Okay, so we're going to have to understand this. How do you make different kinds of actin filament organization in different places and in a dynamic way? And of course, yet again, in micro... sorry?
Is that a cell?
This one is a cell. This one is a closer view of a scanning electron micrograph. The cell would be much bigger. Okay, so that's also an enterocyte. Sorry. So these are microvilli, and what I like about this image is it shows you in the microvilli, so you can actually recognize actin filaments. It's one of these little fibers is an actin filament. They are all parallel, right? So you have literally a bundle of actin filaments. You look deeper in the cell, it's connected, so there's the same filaments contribute to the microvilli and are embedded in now much more of a meshwork. Okay, so again, saying the same thing over and over again, we have the same actin contributing to different organizations in different places in the cell. Briefly, there are a couple different genes encoding actin molecules, classically referred to as beta, alpha, and gamma actin, with some specialization. For instance, the main actin we're going to talk about is beta actin. It's found at the lamellipodium of migratory cells, as well as the cortex, usually involved in these meshworks, these dynamic meshworks we're going to talk about over and over again. Gamma actin is more found as these stress fibers, and alpha actin, you may hear about this contractile ring when you discuss cell divisions, right? So in cell divisions, at the end of mitosis, you need to squeeze the two daughter cells, to separate the two daughter cells, and it involves a contractile ring of actin here. Okay, so that's it for kind of a broad view, and hopefully now you have an idea of the kinds of questions we're going to have to be able to address, right? Especially with regards to polymerization and how it's regulated and how it creates these different forms of cytoskeletal structures in the cell. But let's look back at the initial, the original subunit actin itself, and actin monomer. So now you had to pay attention from the beginning, but now is when you really have to start to be awake and pay a great deal of attention. Okay? So because there's a lot of fundamental features here. The first one is it's a globular protein. It is not a filamentous protein. That means the only way it can make filaments is through polymerization, by adding molecules on top of each other. Okay? Another feature which I always like to emphasize is it's intrinsically polarized. So I don't need you to, of course, remember all kinds of details about the structure, like this 1, 2, 3, 4 sort of subdomains, but this cleft here. So it's an ATP binding protein as well. Sometimes less known about actin, but it can bind ATP. And here, a magnesium atom is represented because magnesium helps stabilize ATP, and that's a general truth. And it has something to do with actomyosin contractility. By the way, let me tell you this, because this is a life hack. If you feel tired and you don't know why, because it applies to me. You feel tired, you don't know why. Like really tired. Your eyes are twitching sometimes. You have like spasm. This is magnesium. You drink a lot of coffee, you eliminate magnesium. If you take magnesium, it helps with this stuff. In part because it helps stabilize ATP, it helps control actomyosin contractility. I'm not joking. It ends the twitching. And sometimes I like, Anyway, it does happen, right?
Yes.
It happens to me all the time. Okay, so now how do we go from this polarized monomer to polymers that will have to dynamically grow and regress and contribute to the formation of this higher order structure, the shaping of entire cells? So people have been studying actin quite a bit, and here is an electron micrograph of an actin filament. And of course, it's hard to see much here besides the filamentous structure and some granularity, right? There's some structure within this filament. Simply put, this is obtained by in vitro polymerization of actin. So the actin, the G actin, the globular actin, when it's bound to ATP, it has to be bound to ATP. If you provide the right amount of salts, it will spontaneously polymerize into F-actin, which, as we'll see, is initially bound to ATP. Eventually, ATP is going to get hydrolyzed. We're going to talk about that. And there's both polymerization as well as spontaneous depolymerization. Okay, so there's an intrinsic dynamic that's just a property of actin. It's actin alone, well, with the ATP and the salts, but there's no other regulatory protein. So polymerization, depolymerization is a spontaneous property of actin. The other spontaneous property which emerges from the structure itself is the organization of the filament. So basically what you have, if you take a close look, is, so you see this thing represents the place where the ATP normally binds. Okay, so it represents the polarity. So I know it looks like little peaches. And you have essentially, it's like you have two strands forming a helix. Okay? And usually these kinds of helices have some mechanical properties that are interesting. We don't need to go into that. The point I really want to make is if you follow this one with the asterisks, you see it's helical, so there's necessarily a periodicity to it. Okay, there's a point in which the same filament comes back onto itself. Okay, and that periodicity is roughly, or half a turn is roughly 36 nanometer. Okay, so it's very important, and I want to emphasize that now because I'm going to get back to it at the end of the lecture. But that means if you imagine the actin filament is a line, and you can only walk when you see a full subunit. It's like you have to walk on blocks that are spaced every 36 nanometer. Okay? We're going to see which molecule does that. You already guessed, maybe. Yeah, myosin. Okay, speaking of myosin, so here it is. So myosin and actin are closely associated, and they've been used together to study actin so as a tool. So here, for example, these heads of myosins have been used because they will eventually bind to actin filaments in vitro. And here the trick is just to use them to look at the organization of the actin, because it provides a bigger structure to look at through electron microscopy. So the point I want to make here is that when people did that, they could see an orientation. So initially, people recognized these sort of arrowheads, right, like this. And that's why the actin filaments have been referred to as, so the two ends of the actin filaments have been referred to as the barbed end, yeah, so that's this side of the arrow, versus the pointed end. They also correspond to what's sometimes called the plus ends and the minus ends. So again, this polarity of the filament is eventually an emerging property of that comes from the properties of the monomers, right? Because if you look at this, let me go back. This one. So the minus ends is here. So wherever the ATP is tends to point to the negative end. Okay. So you have almost a transfer of polarity from the monomers to the filament. And as we'll see, that polarity is very important because you have different kinds of dynamics at the plus end versus the minus end, in particular polymerization versus depolymerization. So I'm going to spend a great deal of time talking about that. Indeed, so that's what I just said. Polymerization happens primarily at the barbed end. I mean, I drew it in this way, but it doesn't really make sense because there's no polymerization in the middle, so to speak. And dissociation or depolymerization tends to happen at the negative end or pointed end. Okay, so those are really important features of actin filaments. This polarity, the periodicity, and the idea that those things emerge from the properties of the monomers. Okay, now let's think about and look at the dynamics. How does it happen? So with actin, this could be studied in vitro. And so what people could do is the following. Is prepare a solution of actin monomers and loaded with ATP. And from what I told you, nothing happens. They won't polymerize until you provide the right salts. Okay, so you can basically, in vitro, decide when you start, when polymerization is going to be allowed to start by addition of salts. So the first, and then what you can do is measure the mass of filaments, in part because if you centrifuge in the right conditions, filaments being bigger, being polymers, they will come down, monomers will stay in solution. So you can dissociate those, you can measure the amount of filaments, okay? So initially, polymerization, right, so the mass of filaments increases, but increases slowly. And then eventually, it picks up, increases much faster. So this first part is called nucleation. And that's a very important step. It's when you have the first monomers that come together. So it's basically harder to go from 1 to 2 and 3 than going to from 2 or more and to just adding filaments. So this next step, which is facilitated, is elongation. The actin filament grows, okay? And then eventually, in vitro, you reach a plateau, and that's called a steady state. And the idea is that you have a stable or steady amount of filaments. That doesn't mean those filaments are not dynamically being made or unmade, right? So there's constant addition on one end and constant depolymerization. It's just that the rates of polymerization roughly equals the rate of depolymerization. Yes?
The salts that you add, do you add it continuously over time, or do you just add it at the beginning?
That's a great question. The total amount of G-actin is fixed at the beginning. So indeed, you probably foresaw that the concentration of monomers decreases from the beginning to when you have the steady state, okay? Because a fraction of these monomers, a decent fraction of these monomers is now into the polymer. So this is why there's this elongation phase, because during the elongation phase, you have basically an excess of monomers, and you have polymerization on both sides of the film, of the nascent filaments, okay? That's a great point. I want to go back for a second to nucleation. So of course, when it's written here, nucleus, not to be confused with the cell nucleus, of course, right? This is just, and there's nothing really special about these monomers. They're not blue versus the other ones, right? It could be any of these monomers. Here is just to say they are the original ones, and what the cartoon is depicting is that at the beginning, you have polymerization on both ends. Again, because you have an excess of monomers in the solution. The next plot is illustrating the idea that nucleation is a rate-limiting step. That is, if to this suspension of monomers you add dimers, trimers of actin, so you add the nuclei pre-made, now the kinetics looks different. You have this rapid elongation from the get-go and quickly reach the steady state, right? So this plot, compared to the previous one, really illustrate the fact that the nucleation step, going from only monomers to some dimers and trimers, is rate limiting. Now, you're scientists in the making, and you like to think about things. So when a step in biology or in a biochemical reaction is rate limited, what does that give the cell? Control. That's exactly right. This was the case with the glycolysis, right? Remember, phosphofructokinase, rate limiting step, this is a point of control. Here, nucleation is going to be a point of control because the cell can modulate that step very well. Not the only one. Sorry, the winter is getting to me little by little, and I'm fighting back. Okay, so to the point that, back to the question of concentration, because what I have not told you is, these experiments I showed you basically assume that you have an excess of monomers in the suspension. So much so that the concentration of actin monomers is compatible with spontaneous polymerization. But like we said, eventually a steady state is reached, and you're not going to polymerize ad libitum, or forever. Now, what this plot is showing, it's a little bit of a confusing plot. It's just showing mass as a function of total actin concentration. So now what you're doing, you're varying the total concentration you use at the beginning, and you're trying to measure, after a while, the amount at the steady state, as a matter of fact, the amount of filaments versus the amount of monomers. Okay? So it's not the same curve. And what this is illustrating is that at low concentration, you only have monomers. Right? So what does that mean? That means below a certain concentration, even if you provide the right salts and even if the monomers are ATP loaded, they won't polymerize. Okay, there's a need for a so-called critical concentration for polymerization, and that is true even if you provide the nuclei. Okay, so it doesn't depend on nucleation. It's just polymerization, addition of monomers to pre-existing filaments. But then above this critical concentration, you see very interestingly, the mass of monomers remains the same, and instead everything you add is being added to the filaments. So this critical concentration is key to decide whether new monomers are going to be added to existing filaments or not. Okay, so let's look at that. Now, this is a neat experiment. So where are they added? Right? Because now we have to comprehend this critical concentration question with the fact that the two ends of these polarized microfilaments are not identical. So in cartoon form, that's, say, a filament we're starting with, and in this experiment, what's done is to start with decorated F-actin using this myosin S1 head domain. I'm going to get back to those at the end, but here we are just using it to identify the chunk of F-actin that was preloaded, that was used to start. Okay? And what the experiment consists of is to then supply actin monomers and see where they go, which side they go. So, because of the barbed end, well, this is the barbed end or plus ends, and this is the pointed end or negative end. And if you add actin ATP, eventually what you get is this. Right? So this is a very striking image, in my opinion. Again, one of those where if I show it to you, you should be able to explain, no. Right? Because it illustrates very, very clearly that polymerization has happened a little bit at the negative end. There's a little tiny bit added here, but a lot more at the plus end. Okay? And of course, in this experiment, you have, by and large, we have the same concentration of actin monomers exposed to both ends, right? So that suggests that either end, they may not have the exact same critical concentration. Okay, let me get into that. So indeed, I'm going to get to this ATP stuff a bit later, but in cartoon form, what's been measured is that at the plus end, you have this addition which happens at this rate, and at the negative end at this rate. And that's the dissociation. So the critical, so people could measure the assembly rate versus the disassembly rate, right? So this is the assembly rate, so that means you're adding, that's the number of molecules you're adding per micromolar of G-actin per second, right? So here the micromolar corresponds to the concentration of the monomers. Simply put, and I don't need you to remember these values, I'll give them to you. I just need you to understand what's going on. But from these measurements, so that's what's measured, okay? You can calculate the critical concentration, which simply put is the ratio of the deassembly over the assembly rate. And if you're not convinced, the units are basically giving it for you. Okay, because the critical concentration is going to be in micromolar. At the end, what matters is the critical concentration here is 0.12 or 0.12, while here it's 0.60 micromolar. So what does that mean? That means that at the plus end, you just have the concentration of monomers in the solution just has to be above 0.12 micromolar for polymerization to happen. Okay? Whereas on the negative end, you have to have a concentration above 0.60. So that's about 5 times higher. Okay? So you have a range of concentrations, typically between 0.12 and 0.60 micromolar, where you're going to have polymerization at the plus end, but no polymerization at the negative end, and therefore rather depolymerization. Okay? So you have, and all of these are properties of actin. So the intrinsic property of the actin itself is providing this ability to polymerize on one hand, depolymerize on the other. Okay? Now there's a very important consequence of this, is the following, and that's called actin filament thread myelin. Because in the cell, and I'm gonna get back to that, because that's the important question, right? Everything was determined in vitro, but one key question is what's the situation in cells? So in cells, the estimated amount of available ATP G-actin is about 0.3 micromolar, smack in between the two critical concentrations, right? So from that, you basically predict that there's going to be polymerization at the plus end and rather depolymerization at the negative end. So that's going to cause this thread myelin, right? And when you have this thread myelin happening, that means you can use polymerization, depolymerization to exert force, right? So you have some movement. If you imagine like the cartoon shows, so this happens in reality, that this blue position is fixed. Just by adding monomers on one side and removing on the other, you're shifting the relative position of the filament. Okay, so this is what's called actin thread myelin. Now you may wonder, well, if there's force, if there's mechanical work, what do we need? Energy, ATP, right? And this is where the ATP kicks in, because it's somewhat coupled, this principle of thread myelin and exerting force and movement just by modulating polymerization is somewhat coupled with hydrolysis of ATP, and that's also polarizing the filament because ATP-loaded actin tends to be added here at the plus end. Hydrolysis happens, over time, and eventually it's ADP actin which tends to be depolymerized. Okay, so you have, if you will, you have somewhat of a coupling between this exergonic reaction of hydrolyzing ATP and the endergonic reaction of actin thread myelin exerting force through polymerization on the plus end, depolymerization on the negative end. Okay? Now, I'm done with my general explanation of everything actin does just based on its own intrinsic properties. And by now, you should already be fascinated, right? Because it's amazing that just by the nature of this protein, you can have all of these basic functions, fulfilled by the polymers. But of course, the cells exert a great deal of control over this. In other words, the intrinsic properties of actin and of this polymerization, depolymerization, they provide a potential that the cell still has to harness and steer towards, establishing the actin cytoskeleton and the dynamics. Yes?
Are the monomers that are like depolymerized, are they like recycled to the
Absolutely. That's my next slide. This is a great question. it makes me feel good because I feel like I've built the narrative so well, you can just know what's coming. Yeah, that's right. Those filaments, right? That was a great question because indeed the actin monomers that leave the filament, they are now ADP bound. And I was telling you they are the ATP bound ones that are added at the plus end. So you need to recycle those. And that's the job of this so-called profilin cycle, which involves this small actin binding protein, which helps take care of the monomers that are now bound to ADP, help with the replacement of ADP by ATP. Now you have ATP bound actin that's ready to go back and be used for polymerization. And this process is essential for the whole system. And now I'm going to walk you through all kinds of examples of proteins that are acting to exert some control over the whole cycle. The next one is polymerization itself, another very important protein, which already tends to associate with the negative ends of filaments, in part because it recognizes ADP-bound actin, and that protein is called cofilin. So cofilin is a small protein, and it's an actin filament severing protein, which basically chops them towards the negative end. And so it does not depolymerize it directly, but it creates more negative ends. So that means now you have, instead of having depolymerization from just one negative end, you have depolymerization from two negative ends, but with the same rate. So by and large, it tends to increase the rate of depolymerization as well, okay? And so that helps with the recycling. And what's sometimes hard to fathom completely is that increasing depolymerization, promoting the recycling of actin monomers, ends up also promoting polymerization. So there is this counterintuitive effect of cofilin, because you could see it as, oh, it's a protein that breaks filaments. But at an integrative level, it actually helps promote polymerization because it helps replenish the pool of actin monomers, right? Because remember the critical concentration. If you don't have enough monomers, you can't polymerize, okay? Talking about that, there's a conundrum we need to solve. I've not told you this, but if you measure the total amount of actin in cells, the range is, the order of magnitude is hundreds of micromolars. Hundreds. We said critical concentrations were 0.12, 0.60. Only about 50% is not polymerized, right? So there's a lot of actin that's mobilized already in actin filaments, but still 50% of this much is, 50 to 200 micromolar. So it's already orders of magnitude more than the critical concentrations. So just from these numbers, what would you expect? Rampant polymerization. Crazy, uncontrolled polymerization almost. Just default polymerization. Indeed, because the critical concentration is in this range. So that's the reaction that is highly dependent on this concentration. So what can you imagine to deal with that? To basically lower the amount of available monomers from this value to that value. What?
The amount of ATP available.
The amount of ATP, that's an interesting idea. There's a simpler solution. Because the amount of ATP can hardly be limiting. I mean, it's used in so many processes. Now, the principle here, like in many other cases, is a sink or some sequestering mechanism. If you hide a large amount of the G-actin from, if you make it unavailable for polymerization, you can actually modulate the amount of actin monomers without creating more actin filaments. And that's the job of a protein called thymosin beta, thymosin beta 4, which is very highly abundant in the cell. See, 500 micromolar. So that protein alone is capable of sequestering a great deal of unpolymerized actin away from polymerization. And so you can now imagine that this third cycle, the thymosin beta 4 cycle, modulates the amount of available actin monomers, and itself is one angle through which the cell can control actin polymerization. Okay? Now, those three are, kind of first level control over actin polymerization, and we're going to see more and more. Right? There's many layers to regulate the actin cytoskeleton. Others include elements that will stabilize the filaments themselves, because, what I should say perhaps is that many of these, coffulin acts on the filaments, but these other two, they act at the level of the monomers. Okay? Now, there's a great deal of control over the filaments themselves, and we're going to see a couple examples. So one of them here is tropomodulin. So one way to stabilize filaments, so you could imagine many ways to stabilize filaments. One of them is to prevent depolymerization, right? So for that, because we saw that at the cellular concentration of actin, most depolymerization happens at the negative end. Well, if you have proteins like tropomodulin binding to the negative end, that's it. No depolymerization. And these kinds of proteins are important in muscles, for example, where you need long-lived, stable actin filaments for the contractility to be possible. Other kinds of regulators that have almost the opposite effect, this capping protein, they will instead bind to the plus end, and so what do they prevent? Polymerization, right? So that's a case where you would want to, instead of stabilizing filaments, boost dynamics, right? So allow short filaments to be created, but then prevent them from growing further, such as, for example, in a meshwork, right? So again, somewhat counterintuitively, in an actin meshwork, you have an important role for capping proteins because they prevent or they sort of modulate the length of filaments. And in this way, they favor the branching, the making of multiple filaments as opposed to just a few very long ones. Okay? So this is regulated in several ways, like lipids in the plasma membrane can modulate this, and gelselin, which works also in signaling with calcium. So gelselin is interesting because it can both cap the plus end as well as promote severing and depolymerization on the negative end. So those are just a couple examples. If you put them in the big picture, interestingly, they will act at different levels, right? So if we go back to this kinetics of actin polymerization and, a steady state maintenance of filaments, you have the profilin and cofilin cycles together with the thymosin beta 4, which regulate the availability of G-actin. You have cap and gelselin, which will tend to block or inhibit polymerization. And I just told you about tropomodulin, which will stabilize filaments by inhibiting depolymerization here. Okay, so you already see that in the different step in the actin polymerization dynamics, the cell exerts control. But which one is the rate limiting? Nucleation. Did I tell you about control over nucleation? Not much. Okay? In a way, the abundance of monomers will impact nucleation, but as you can anticipate, there's a great deal of direct control over nucleation, especially by promoting it, right? Because nucleation being limiting, if the cell develops mechanisms to boost nucleation in a controlled way, in defined times and places, it's going to basically be able to choose where and when to start the making of filaments. Okay? So we're going to look into that. So indeed, So back to this, I'm gonna building kind of a smooth transition here, but everything has to do with this organization of the cell, right? So you want stable filaments here, you want more there's quite a bit of stability in polarized cells. We saw this before, and different organization in microvilli versus the cortex. So we're gonna talk about that and the end. So I'm going quickly through that because I'm just so eager to get to the point, and you're going to understand why. One of my favorite proteins coming. So that's for the epithelial cells. In migratory cells, you have to have different organization, different dynamics in different parts. Filopodia with longish filaments, kind of bundled. The leading edge with a meshwork, so that means shorter filaments, not bundled, but instead forming, a meshwork. Stress fibers, long-lived, bundled, and then more cell cortex, a bit less dynamic than this one. So if you think about it, you could already start making hypotheses about which ones of the activities I showed you before, right? The tropomyosin, for example, the stuff that will stabilize a filament. Where would you expect it? Here. In the stress fibers, right? So that's the kind of connections I'm trying to make you make. Good one. But those don't regulate nucleation. What regulates nucleation? Signaling, right? So this is a very conceptual slide in some ways, but the idea is that the cell, so a migratory cell, and we're going to see more precise examples of this, will migrate in response to attractive cues or chemorepellent cues and so on. So those are sensed. So this is information from the outside, from the exterior of the cell. It needs to be sensed, and that's usually the job of transmembrane receptors, and relayed to the cytoskeleton eventually, right? So a great deal of control happens here in this signal transduction pathway. So we have to identify proteins that on the one hand will be controlled by these signaling inputs, and on the other hand will directly modulate the cytoskeletal dynamics. Okay? So now we are going to start looking at the most important slide of this entire course, right? So pay attention. The good thing is you've already seen these kinds of proteins. It's again a small GTPase, except it's the row family of small GTPases, and this is how they work. So they are normally in an inactive state, very inactive state. They are not only bound to GDP, but they're also held in the cytosol through this GDI, GTPase dissociation inhibitor protein, which basically helps hide this farnesyl-farnesylated moiety. So that's a lipid modification at the C-terminus of the protein, which normally helps anchor it to the membrane. So one step regulating this protein is this anchorage, or this anchoring, whatever the right English word is, of this protein to the membrane. Anchorage is a city, right? So it's more like the anchoring. My bad. Excuse my French. But it's still GDP bound. Okay, so GDP bound means inactive. That's the same for RAN, that's the same for RAB, that's the same for SOR1, that's the same for OFF. This is this giant superfamily of small GTPases. Their activity is regulated by being bound to GTP or GDP, but that's all about conformation. It's not, they don't work by hydrolyzing GTP. For them, hydrolyzing GTP is what inhibits them, is what regulates them. And so indeed, what's going to activate those row GTPases are GEFs, guanine exchange factors, just like the other ones. But those are the ones to be more or less directly in contact with the signals from the outside. So here you have a first connection between the signaling from the outside via the receptor and a guanine exchange factor, or guanine nucleotide exchange factor, that will activate a row GTPase, right? And then to complete the cycle, and we're going to see specific examples, once activated, so it's at the plasma membranes. That's a very important point, right? Because this is where you're going to have a lot of the actin dynamics being regulated just underneath the plasma membrane. And in part because this is where the Rho GTPases, the active Rho GTPases are, right? So you have in direct contact with signaling. So once they change conformation, right? You see this little thing sticking out now, they can interact with the effector protein. So it's all about protein-protein interactions and conformational changes. And then eventually, they go through the same cycle we've seen before. They are inhibited by gaps which promote GTP hydrolysis and the release of inorganic phosphate. Right? So again, this is a very important concept, and every year I say, if there's one thing you walk away with, this is it, right? So no excuse. If it's on the exam and someone after the exam comes, oh, but I did not. No, this stuff, you know it, okay? I have to be tough a little bit. Okay, so let's see a couple examples, right? Because the one thing I've not told you yet is how this relates to actin dynamics, and that's the key question we have to have. So I'm just going to walk you through a few specific examples, and oh, by the way, what part of the actin dynamics do you think these guys will regulate? Nucleation, right? So don't lose sight of this. It's a key rate-limiting step. Now we have the beginning of a handle on this step. And like I told you, because this step is unfavored, most of the regulation is about promoting it. Okay? So those regulatory proteins, we'll see they tend to promote nucleation, they tend to promote actin filament polymerization. So the first one I really will talk about are these formins. And they do promote both nucleation and elongation. And you'll see why I say here of unbranched actin filaments. So they have all of these effector proteins. So now those are effectors, right? So they are the ones that are themselves being regulated by the Rho GTPases. So they all need this RBD domain, which stands for Rho binding domain, right? So there's one domain to bind to Rho, and then there's a formin homology domain 1 and formin homology domain 2. So what do they do? So what they do is they kind of dimerize, and as dimers, they bring actin filaments together. So that's it, right there. They are, just by their structure, once they are activated, they stop nucleation, okay? Because they bind actin, and then once they are bound to the first dimer, they actually remain bound. And they remain bound on the plus end, but they have conformational changes whereby they somehow allow for the addition of new monomers, right? So, which we saw normally happens spontaneously. So all they need to do is they allow for the addition of new monomers at the plus end, but by being bound there, they basically prevent capping. Okay, so they have this so-called anti-capping activity. And what this image is showing, so this is a nascent actin filament, and this is a formin. Well, it's recognized by an antibody with a iron or gold particle, sorry. So the formin is bound as the plus N, right? But so there are two key aspects of formin activity. They promote nucleation, and by remaining at the plus end but allowing addition of new monomers, they prevent capping. Okay, so that's why they promote both nucleation and elongation. So of de novo filament, they don't need a template, right? They can just help nucleate from scratch, well, using monomers, and then promote elongation. And the reason why they are regulated by Rho is because formins are normally auto-inhibitory. So they fold onto themselves, and the N terminus here, where the Rho binding domain is, tends to interfere with the ability of these FH1 and FH2 domains from binding to actin. So the whole story is when Rho is activated, so that's downstream of signaling, downstream of GAFs, it can recruit formins, and by binding to the Rho binding domain, relieve this autoinhibition, which now allows the other formin domains to nucleate and promote actin filament elongation. Right? So now you have all the steps whereby this signaling GAF Rho formin pathway can start nucleation and promote the formation of new filaments. Okay, and that's very important. We're going to see an example of this when we talk about cell migration. Now, this would create filaments, individual filaments, right? So this could be useful if you think about the other previous cartoons when you need a certain length, for example, and so on. I mean, you need to create new filaments in many cases. But when you have a meshwork, I was telling you, sometimes you need shorter filaments and so on. And as a matter of fact, when people took a close look through electron microscopy, now this is the actin meshwork of a lamellipodium. I think I have to speed up a bit. We have these branching structures. And this branching always happens where you have almost like a mother filament or a template filament, and another filament sticks out from it at about 70 degrees. Don't worry about the 70 degrees. This can be recapitulated in vitro. So here you'll see, and if you take a close look at this movie, you have some places where a filament is born, but then eventually another filament sticks out from it, right? So, and it's recapitulated in particular using this complex called ARP23. So ARP stands for actin-related protein. These are proteins that resemble actin. And they tend to bind here on the side of actin filaments. And normally, although they resemble an actin dimer, they still need to be activated, so to have their conformation changed by activation through this protein called WASP. And when this happens, now you have this template, so these two monomers, and they look very much like a nucleus. Right? So right there, this can now serve as a template for polymerization to add actin monomers. And that's how you start making the new filament. Okay? Yes? Yeah, the ARP23 complexes, they tend to associate with filaments, kind of by default, but they won't promote the formation of a new branch until activated by WASP. Okay? Now, what do you think regulate WASP? Rho GTPases. Indeed. Especially one of them here, CDC42, which we'll see is important for polarity, for migration, and so on. And so WASP itself has a kind of a similar logic as the formins. A Rho binding domain for regulation by Rho GTPases, and then a effector domain for interaction here with the ARP complex. And actually even recruitment of monomers. So CDC42 is the Rho GTPase, activates WASP, feeding onto ARP23, responds to a variety of signals and GAFs, and you even have another branch, also using other signals, other GAF. Again, there's a great deal of variability and tissue-specific activity here. I have no time to go into much specific. We're going to get back to this when we discuss migration. But RAC is another important GTPase doing this, acting through wave and also promoting ARP23-mediated actin filament branching. Very briefly, I just want to illustrate how certain pathogens harness this property. So here what you see are bacteria inside the cell moving. like here. See these crazy movements, right? So listeria, it can enter the cells and then move around. And what they do is that they have, on one side of the cell, of the bacterial cell, they have a molecule that mimics wasp. And so they basically promote actin polymerization behind them. So and they use the force of actin polymerization to move around. And indeed, in fixed samples, you can see bacteria here in red. And if you stain with phalloidin, that molecule which recognizes F-actin, you see these comet tails or actin tails here. And that's indicative of polymerization behind the bacterium and triggered by the bacterium. All right. So I have to speed up a little bit in the rest, so I can tell you about myosins. The next couple of slides, I just want to illustrate that although a lot of the regulation has to do with polymerization and the dynamics of actin, so that's most of what I told you about so far, there is also control of the organization of existing filaments. And I'm just going to give you a couple examples. If you think about bundles versus network, and if we forget about the dynamics of polymerization for a little while, well, you have dedicated proteins to define this architecture. So fimbrine, for example, or facin, which I'm not highlighting here, can bind filaments twice, and therefore they tend to help bundle them. Other kinds of proteins, like filament, instead, one common feature, they all have to have two F-actin binding domain to bring two filaments together. But then eventually the structure in between those domains will define the kind of structure they promote. So here, this one, filament, would stabilize actin meshwork and so on. Okay, so in the time left, I'm going to tell you about myosins. Sorry, it's going to have to be a bit fast. But I really need to tell you about myosins. So myosin filaments were studied extensively in skeletal muscles. They make these polymers, and they are directly involved in contractility because of their ability to use the energy of ATP to exert force by binding to actin. Right? So these are essentially the key properties of myosins that you need to worry about. And this comes from their structure. So you have typically two myosin heads attached together by their tail, and that's the heavy chain. So this is one protein, and this is one other protein. That's the key structure of myosins. The business end, where you have both the ATP domain here and the dimerization domain. But the activity is going to be regulated by these so-called light chains, which tend to bind at the neck region, and they can be regulated themselves by phosphorylation. And I'm going to tell you about that when we discuss migration. Just for illustration purposes, the head domain is extremely important because this is where you have the nucleotide binding site. So that's the ATPase domain. That's what's going to harness the energy of ATP. It's also the domain that binds actin. Right? So now you have a motor that can be fueled by ATP and bind actin. So use actin as a track. Okay? And again, these other chains are regulatory. I'm not going to tell you much more. And the tail helps to tether the myosin to cargos and so on. So this could be used, in vitro. So you can take myosin and cleave it using peptidases in some ways, attach it to a substrate in vitro, and then provide fluorescent actin filaments. So why am I telling you this? Because this was one way to measure the properties of movement of myosin. It's going to be clear when I show you these images. So let me just show you these three at once. So these are three snapshots of a time series. the same actin filaments, short actin filaments attached to myosin in vitro. And if you look at this one here, one starts here, then moves in this way, and moves in that way. And 3 also moves upwards. So this is all representative of those myosin heads. They are attached to the substrate, but they can still exert some force. So what they are doing is they are moving the actin filaments. The actin filaments here are not polymerizing at all, right? They are stabilized. They're just being moved about by the myosins. Why am I telling you this? Because this could be used to measure properties of myosins as motors. And one of the key aspects here is that if the speed is measured, you see it increases with what? With the length of the neck. So it told something about how myosin works, and in a nutshell, it's a lever arm principle. In other words, if it grabs actin on one side, then lets go and does this. Okay, so you imagine that if you have a longer arm, and if you have the same rhythm, you're going to have more movement, more displacement in the same time. Okay, so that means the rhythm is the same, but the length depends on the structure, the length of the neck. Other kinds of measurements done with one head of myosins led to the building of this kind of graph. So here what you're looking at is over time, the displacement. And what I really want you to get from this is it's stepwise. So it's not a continuous displacement, right? So if the myosin was somewhat gliding, you would have a continuous curve. Here, stepwise is also consistent with the lever arm. In one position, it go through steps. And interestingly, those steps are now 36 nanometer. Why did I tell you about this? It's the periodicity of actin. Okay? So this model that if the myosin is sitting on one block, the next block it can step onto is 36 nanometer away, and that's in part because of the property of myosin, but also because of the property of actin. So the two have kind of co-evolved, if you will. Okay. Now this is with one head, and if you do this experiment by using an entire myosin but labeling only one of the heads, and now the measurement shows you it's 72 nanometer. Okay. What does that all mean? This means that when you have two heads, it's this so-called hand-over-hand movement. In other words, it's attached here and here and moves over and over. And you only see, so the same head does steps that are twice 36 nanometer, right? And here you see only the movement of one head. Okay, so this is all consistent with myosin basically walking on actin filaments with alternating steps of 36 nanometer each. So that's what this cartoon is representing, and nicely showing that here there's binding to an actin monomer, and the next one in the same conformation on the same line is 36, actually in this case 72 nanometers away. Okay? So this is all built in the structure. Now, I made this slide to sort of illustrate, it's kind of a complicated slide, but what I want to illustrate is the coupling between hydrolysis of ATP, right? Because we can't forget that this force, this movement is powered by ATP hydrolysis. But of course, the coupling has to do with changes in conformation. So this is how it goes, and don't freak out because I don't need you to memorize this. So, but let's say the first step is hydrolysis of ATP, and that causes this so-called head cocking, or this change of conformation here in a myosin head that's not bound. Then this conformation now favors binding to actin. Now you have myosin ADP plus PI bound to actin. This itself favors release of inorganic phosphate, and that's the power stroke. So it basically returns to the original conformation, and that's where you have actual movement. So the reason why I did that is to show you that you have a bit of a decoupling. Well, fundamentally the two are coupled, but they don't happen at the same time. Hydrolysis of ATP happens here. It causes a change of conformation, and then the movement happens in a third step, right? So if you will, this change of conformation is somewhat thermodynamically unfavored and contains energy, if you will, okay? For short, you have energy hydrolysis or ATP hydrolysis and the movement happening in slightly different step in the whole cycle, right? And then of course the whole cycle finishes by replacement of ADP by ATP. There we go. And to finish, and then the head leaves actin and we're back to the original state. So I'm almost done, 3 minutes left. So that's good, because for the last slide, what I want to show you was this movie, was because you could still say, well, this is all kinds of very indirect evidence, and this idea of a molecule walking on an actin filaments give us a break. Except since then, people have used atomic force microscopy, which allows to look at very high resolution, both spatial and temporal, at molecules. So here this is in vitro reconstituted actin filaments, and you're going to watch myosin molecules basically walking on them. This one is probably the clearest. No. Come on. All right, now you believe me? Okay, I'll stop here.